Yep.
Perfect. Oh, wait, you went back out of presenter mode. There we go.
Excellent. Thanks for the lovely introduction, Arwen. I feel now really very much under pressure, but I will do my best. So today I will talk about a bit of a side project that became quite an interesting side of our research, and that's the combined spectroscopy and scattering to resolve biomolecular processes. So rather than being a very technical talk, I've sort of put together a story about how I got to that place and what we think is the future for this type of technology. So the outline is relatively simple because it's a short talk, and it's a little bit of the motivation of why we are doing what we're doing, basically. Some introduction to the technology, so multimodal characterization, and then a couple of examples, and then some of the challenges ahead for this type of technologies. So in my group, we work on basically soft and complex biomaterials, and the research focus is very simple. It's basically how materials works. And we have three pillars. One that is the natural materials, so hierarchical materials, basically saying hydrogen bonded networks, and these elastomeric materials that I will introduce in the next slides. And the things that we're interested is how are the prerequisites to manipulate those materials and leading to functional hybrid materials. But what we have found out through the years is that for those materials, we need to develop in parallel sample environments. And that will be a little bit the focus of this talk. How do we get to this need to develop more and more complex sample environments? And the central question, because we still have science behind us, is really how structural organization emerges and governs macroscopic behaviors. So how did it all start? So as Arwen say, I work on silk, and silk has always been really a driving force in our research, both for scientific questions, but also for instrument development. And one of the key challenges is really to understand how the animal turns a solution of proteins into high-performing fibers. And this is what we call the elusive spinning process. So generally, silk is not alone in this family of elastomeric proteins. So we have silk, of course, center of our interest, but you find these elastomeric proteins in all sorts of realms of the natural world. So for example, when we bake bread, the inside of the bread that makes it very fluffy and elastic is also an elastomeric protein, the high molecular weight gluten, but also in connective tissues, the resilins, so the elastic aspect or the elastic contributors to the insect, allowing them to jump very high, casein, again, muscle foods in the sea arthropods. So all those share common characteristics. Their folding is a bit unclear. So they are neither unfolded, neither denatured. And they all have this modular design. So if I take the example of a spiders, where we have a two-dimensional web, radial threads, and then spiral threads, the yellow are the genetic markers for elasticity, so very elastic-like domains, and then the red threads are more the crystalline type of domain. So you can clearly see from a genetic point of view that the two materials have very different already molecular making, one being very elastic in the spiral threads, and one having a bit of a distribution of these elastic and non-elastic domains. But what makes it quite interesting is that they are neither proteins nor polymers in their behavior. So we have to be right in the middle of the two fields to be able to understand how those interesting proteins behave. So research on silk has been happening for quite some years. So there is a fair amount of knowledge on how the animal takes the protein, secretes it, transports it, and eventually spins it into a fiber that has all sorts of mechanical properties. But here what is interesting is the length scale. So we are talking about small, a few angstroms to micron to millimeter sizes. So all of it is under control and really well orchestrated to allow the animal to get to these final properties. And this has been really one of the challenges in the research, which is what is then the nature of these seal proteins in solution? What is it that the animal is doing to this very complex system so that it can manipulate and eventually get the macroscopic properties of interest? And this may not sound very difficult as a question because one says, okay, it's just a protein. But here is a typical sequence of a silk proteins. So it has about 5,273 amino acids, and there is a strong bias towards the glycine and alanine. About 90% of the amino acid is those two amino acids, which means that if you were to plot how the glycines are being populated on the Ramachandran plots, you will see that this protein can literally explore conformation all over the place. So one question is, how do we then resolve these structures and interactions in complex materials? And this is where sample environment becomes interesting. So if we were to take the simple approach of using one method, we may be missing some aspects. And this is why we are now living in a subject-centric world where we need integrative techniques. And this is a rather famous illustration, which is the blind man and the elephant. And what it says is simply that if you are taking different aspects or different parts of the elephant, you might actually see or interpret different things. And there is a need then to bring all those aspects together eventually to understand that this is an elephant. But one of the challenge that we have is how many techniques do you have to put together? Because you could run the risk of having 6 blind men looking at other 6 blind men. And this is the whole challenge of the research. How far do we need to go in the integrative biology, so the multimodal aspect of the techniques? But first, we have to define what do we need in terms of biophysical method. And after some thinking, one could sort of sit down and write this as an ideal biophysical method. So it would have the capability of observing atomic level structures and dynamics of biological molecules in vivo. And it will also permit the visualization of the structures that form throughout the course of conformational changes or chemical reaction, regardless of the time scale involved. So there is a lot of constraints into that. But if we look carefully and take this type of information from the scattering scale, we could see and map different techniques. So let me walk you through this particular diagram. So this scattering scale in the middle here is basically the type of data that you will get from small angle scattering pushed towards diffraction. So you would see the shape and envelope, the fold, and eventually the atomic structures. And in terms of techniques, you will have the crystallography, you could have optical spectroscopy, small angle scattering, and if you want to go to higher length scale, you have light microscopy, hydrodynamic. But one should not forget that you can also have quantum mechanics on the other side here. So our idea was then, can we use just this side of the static information, so by mixing small angle scattering and optical spectroscopy, to try to resolve those particular interactions. And this is how the multimodal spectroscopy adventure started. So nowadays we're talking about the SURF and the NURF because we have gone a few miles since then. And the SURF stands for SAXS, UVVs, Raman, and fluorescence. And the NURF stands for neutron scattering, UVVs, Raman, fluorescence, and density. And the SAXS here is with X-rays and of course the neutrons with neutrons. And depending on the facilities, then you could see that in neutrons, there is also an interesting need to have density, but I will not talk about neutrons in this particular talk. So this is a timeline of what we've been doing. So we started about 2012, 2013 at the Max 4 with Thomas Plivelich, and with some bits and pieces that I had in the lab, we were able to put together a sample environment where we had a capillary to do the X-rays, so the standard SAXS, and then on the sample holder, we could do the Raman spectroscopy and the UV. Then after that, we moved to a second generation with more probes, fluorescence, Raman, UVVs, and X-rays, and eventually a third generation that was developed in collaboration with the swing beamline at Soleil, where we had a more tidier setup with the same idea of fluorescence, Raman, UVVs, and small angle scattering in these directions. And in parallel to that, we've started also to work on neutron scattering. But the whole idea is always the same, being able to match those two length scales. So the one that we get from the scattering and the one information that we get from the optical spectroscopy. So why would you do that? So the first things are what we call the technical goals. So we want to be able to measure simultaneously the same sample volume. So basically, when you take a UV spectrum or fluorescence spectrum, you want to know that this is exactly the same sample that you have measured. It seems trivial, but sample preparation is really tricky because you have to be able to say, yes, I have been able to repeat exactly the same experiment using two instruments. Here you have the immediate gain of having the two information from the same sample volume. But also as a diagnostic, is the sample I started with the same? And then eventually you can start driving some of those samples into particular direction. And this is what we call control perturbations. You can heat, cool, chemical, mechanical flow, you name it. And then eventually we also thought about the portability, because this should not be a sample environment that is just for the select group of people, for a select group of people. This should be relatively democratic, something that is found at literally any beamline, so that you come and then you get it as a free set of information from getting your SAXS, for example. But what is, again, very important is the scientific outcomes. So can we now, with the integrative approach, talk about component resolution? So looking at a kinetic or a process change and say, yes, we can identify uniquely two components that have been involved in this process. Can we map features to structures, meaning that a peak or a shoulder, does that correspond to a particular structure, meaning a beta sheet or an interactions that we have in our systems? And can we look at more complex things, for example, cooperative processes? So now I will try to give some light-hearted examples about some of those applications. And once again, the challenge and the question is when and where interactions happen. So in the first example that we have been able to do is looking at human serum albumin response to oxidative stress. So there is a long-standing question about hypochlorite-induced oxidations. And recently, there was a paper in 2021 where they could sort out the hypochlorite partitioning, that albumin could take some of it, fibrinogen could take some of it, but the activation of the albumin could lead to very severe problems, whereas the fibrinogen will be less problematic, so will act more as an antioxidant rather than the albumin acting as a cytotoxic. So as I summarized to the side here, this is a known effect. It has been widely measured by individual techniques, so there have been ways to identify and quantitate these oxidative stresses, and the negative effects, as I say, are very well known. But there is no understanding of the mechanism and the role of the albumin, and in this case, we're interested in the human serum albumin. And interestingly, also, what makes human serum albumin a reactive species scavenger? So what we set to do, and that is really the work of Alessandra Del Guidice, now in Rome. She visited the lab for 6 months, and then we just worked on this particular integration of the human serum albumin induced oxidation using SAXS, UVVs, and fluorescence. And this is a bit of a busy slide, but basically with the SAXS, the small angle scattering, you get an idea of the shape as you titrate the proteins with the hypochlorite. But since you're doing the UVVs simultaneously, you also get some effect on the tertiary structure. So you see some shifts in the UV absorbance, and then in the fluorescence, you also see some quenching as well as some shifts. And what was interesting is the combination of the three techniques allowed us to put together an hypothesis that the human serum albumin acts actually as a structural buffer for the hypochlorite-induced oxidation. And one could deduce unique shape and forms for this human serum albumin as it is being titrated by increasing amount of hypochlorite. So there are few other works that we've done with Alessandra looking at acidifications and other things that also illustrate the particular aspect of combining the techniques. And here, I would like to come back to this particular comment of how many techniques do we actually need? And there is mathematically a theorem saying that if you can have at least three independent techniques, you can resolve what's called rotational ambiguity. So when you try to derive components or number of components from a process by having three independent techniques, you can be ensured that your decomposition will be unique. And this is why we are really aiming for all the time when doing experiment to have at least three measurements. So the UV, the fluorescence, and the scattering in this case. So another aspect that has also interested us is how clean is the process. Because most of the time when you prepare your protein, you're never certain that you have exactly a particular that it is in a monomeric form or that it has a few dimer or it has aggregates. And that has been highlighted quite recently in small angle scattering by hyphenating chromatography. So there is nothing new with inline chromatography in scattering, but what we wanted to do was go a little bit further, because you would say, okay, I can separate, I can have some interesting information about my proteins and the monomeric form of it, but that's it. So what we decided to do was to add what's called a reaction loop. So you would do your separation as you would normally do. So you will have an HPLC with a size exclusion column. But what we do instead, we have devised, and this is actually work done in collaboration with a colleague, Per-Olof Larsson, who's an expert in chromatography. So we built a second chromatography system where we could continuously pump a substrate for this enzyme, the beta-galactosidase. And as we chromatograph the beta-galactosidase coming from a technical grade source, we then mix them with the substrate and measure the reactivity as well as the structure and the chemistry of this particular enzyme. And this is how it looks like. So the first thing you get is, of course, your trace. You will have the UV spectrum of your beta-galactosidase being eluted. And what is interesting is that you can now overlay the activity, so the conversion of the substrate into a product that correspond to this elution profile. And here you would say there is only one big fraction and maybe a shoulder here. And what happens is that there is a big fraction that has a certain reactivity, but also the shoulder has also some reactivity. So if you were to do and take this particular enzyme and measure it immediately, you will have a mixed reactivity from the main component and something else. And then, of course, you could look at the fluorescence. So here it's only the trace of the fluorescence emission maximum, but for each of the frame, you will have a full emission spectrum. So you can look at how the solvent or the environment is changing for this particular sample. And then, of course, you get the scattering intensity. So here we'll just plot the scattering intensity of zero angle, which is in a way proportional to the molecular mass. And what we see and confirm is that there is a large entity here, and then of course there is the major entity. The major entity has the highest activity, but the larger entity also have some contributing catalytic effect. So without going into the details, what is interesting here is the general questions that you could ask with such an approach. So you could ask, what are the fractions? What fractions are catalytic? And that could be quite interesting, again, when looking at technical grade enzyme with maybe similar enzyme being produced in the in the pot, or if you have multimeric enzyme like the beta-galactosidase, is it active as a monomer, is it active as a dimer, or is it active as a tetramer? So for the beta-galactosidase, for example, we know that it's only the tetramer that is active. But also in the same study, we looked at how and what happens under competitive conditions. So in this case, we added some galactose and then some lactose and try to see how are those two different substrate changing the catalysis and changing the UV, the fluorescence, and the scattering. But if we go further than that, we could also ask the question, can we use this particular approach to see how protein could be formulated, meaning testing some excipients? So I will have a chromatography where I have only my monomeric system, and then on the parallel channel, I will run some excipient to see how would my molecule behave if I add, let's say, a lot of sugar to protect it or any other excipients. And then the list could go on forever. But here we ended up with an interesting challenge, which is, can we automate further? Because a chromatography run takes quite some time. It takes about half an hour to an hour, depending on the resolution that you're interested in. And then you still have to do some injection, you still have to do some preparations and sampling and etc. So we asked the question, could we then do it all automated? And this is what I call chromatography and beyond. And this is quite an interesting aspect when it comes to the next generation synchrotron and the next generation spallation sources, because we're talking about instruments that have an extreme high brightness, so designed for high throughput samples. So we are now running in the situations where we don't have enough samples actually per shifts of the instrument. So we need to increase the way we prepare the samples. So in this particular case, what we have then taken advantage of is what we call the step purification methods and using some loops. So the first generation that I've introduced was simply taking a standard HPLC, single column, our parallel substrate, and then the reaction mixing loop. But now we could use, again, similar ECTA systems or chromatography system, but use what they have and call the loop reaction valves. So basically what you do, you run the HPLC once, you identified where your fractions of interest are, and then you just run it several times. And then what you do, you store your loops in the loops, the fractions of interest. So for example, our beta-galactosidase main peak, we could save it in several peaks. And what we do then is just automate and say we would like to inject any of those fractions now with our reaction loop and look at the reactivity. So all of a sudden, we can now automate and do 5 or 10 experiments with the same sample without having further interactions with the instrumentation. And this is how it looks like on the ECTA setting up device. And this would be what I would call the ultimate experiment, where you would combine the separation, the loop fraction collection, and then the post-column reactions, because eventually you want to know what happens to your sample when you are transforming it or when you are reacting it. And then, of course, it goes into a SURF or a NURF, where you have a single set analysis, component resolution, correlation analysis, and etc. So rather than bore you with all sorts of data, I would like to present mainly a current status and some of the next challenges that we're facing in this particular endeavor. So the first one is a current status. So now we've been really pushing for having this technology as democratic as possible. So with a collaboration between Soleil and Max4, there is now a SURF available at the swing beam line. So when you apply for beam time, you can tick SURF, and then automatically when you collect your SAXS data, you will get the UV and the fluorescence for free. At the Max4 and the COSAX, when the instrument will be up and running, we will also have this particular instrument available for users. And in parallel, we did that for the neutron. So now at ISIS, at the Larmor beam line, you also have the NURF available when applying for beam time. And others to come are including the ILL and the FRM2 for the neutron sources. But we are still having some difficulties with data management and visualizations, because all of a sudden we are producing a large amount of data, time-stamped, that needs to be visualized very quickly so that you can anticipate and decide which experiment you will run next, but also have a quick sort of check and see, is everything okay or not okay? Then there is a big effort at the moment where we're trying to up this integrative approach. So how do we write better codes for component analysis and correlations, rather than saying, okay, something is changing in the scattering, something is changing in the UV, something is changing in the fluorescence? Can we actually weigh each of those information sources and make a more coherent and integrated data analysis approach? And then, of course, there is interest with the viscous samples, because at one point, we cannot just work with low viscosity systems. There is the integration of other modalities. So with the X-rays, we've been successful at integrating Raman to look at, for example, carbon nanodots and carbon dots, and then light scattering, but I'm not really a big fan of light scattering in that perspective, because it's extremely difficult to implement, which goes a little bit against the ethos that I've been trying to sell here, which is the ease and the portability of the system. And then, of course, this liquid handling system, because now with those high throughput system, we need to be able to up the sample preparations to address this particular bottleneck. And eventually, something very selfish, after a few years, I will eventually be able to do some silk experiments, because so far it has been a bit of a challenge, either because of radiation damage or the viscosity of the sample to be transported to the visualization port. Of course, a lot of people to acknowledge, and I think the list is very long, but I've tried to summarize it, because a lot of the work couldn't have happened before without the help of the Max Lab laboratory and Thomas Plivelich in particular. And then some of the collaboration with the swing beamline at Soleil with Javier Perez, and in Lund with my colleague Per-Olof Larsson, who really took the chromatography to another level. And then, of course, Alessandra in Rome, that did all this nice work on the human serum albumin. So you can check all the papers that we have out with that. And then now with the neutrons, a collaboration with Adrian Rennie and Adam Engberg in Uppsala. And then, of course, with the people at DSS, Andrew Jackson, Judith Houston, and Anders Pettersson. And then at ISIS, of course, Rob Dalglish, Sarah Rogers, Andy Church, and at ILL with Lionel Porcar and Anne Martel. And now Christina Cocho, who is helping us with the data visualization and integration. And then, of course, all this would not have happened without some fundings and some really good friends that were really, how to say that, kind to me when I was babbling and trying to get stuff out of them. And thank you very much for your attention.